students, welcome to today's video. In this video we are going to see in details about CTAB plant DNA extraction. So let's begin. First of all, we are going to see different chemicals that are involved in plant DNA extraction, and then we are going to see role of each chemical, and we are also going to see what are the instruments. Okay. So first of all, let me discuss how the DNA extraction in plants is difficult as compared to DNA extraction in other microorganisms such as bacteria, fungi and animal cell. Okay. So first of all, let me discuss how it is different in plants as compared to microorganisms in animal cells. Okay. So what happens in plants is, first of all, there is a presence of cell wall that is made up of tough material, that is cellulose. Yes. Plus, along with this cellulose, there is presence of cell membrane and other structural polysaccharides are there. Polysaccharides are present. Then polyphenols, tannins, and all other compounds like this are present. Okay. So what happens? These compounds does interfere in the DNA isolation process. Okay. So therefore, the DNA isolation in plant cell becomes difficult. Okay. So there is an efficient method for the isolation of DNA in plant cells, that is CTAB plant DNA extraction. Okay. So let's see in details the method, that is CTAB plant DNA extraction, and we are going to see different instruments here. So first of all, there are different instruments. First instrument that is required is centrifuge, cooling centrifuge. Then we require water bath or dry bath. Then we require mortar, pastel for crushing of the DNA. So there are different instruments required here. So let's see now the chemicals that are required in CTAB plant DNA extraction and its preparation also. Meanwhile, I am also going to tell you the roles of all the different chemicals that are present in CTAB plant DNA extraction. Okay. So first of all, the first chemical that is required is liquid nitrogen. Okay. Okay. So what happens, first of all, for the isolation of DNA, you have to grind the plant sample. Okay. So when you grind the plant sample, no matter how finely you grind it or how finely you homogenize it using mortar pestle, you cannot get the consistency or the fine texture of the homogenate that you get through liquid nitrogen. Okay. So what happens, liquid nitrogen is having a temperature of -196 degrees Celsius. Okay. So what happens when you put liquid nitrogen in a mortar pestle and you put leaves of plant from which you want to isolate the DNA into that, when you put liquid nitrogen on this, or when you put leaves in the mortar pestle taken with liquid nitrogen, immediately whatever the cells that are present in this leaf are having water in it. Okay. So you know the water is present in all living cells. Okay. So this water, after the addition of liquid N2, gets crystallizes immediately. There is a crystal formation takes place. Okay. So due to this, what happens? The crystal forms immediately into the cell, inside the cell. Okay. Now these crystals are sharp. Okay. So what happens when you crush these plant cells? These sharp crystals tend to make a powder of that leaves or the plant tissue that you are taking. Okay. And the homogenate that is formed is super fine. Okay. So the homogenate that is formed here is super fine one. And one more important thing is the DNA, whatever that is formed here, is intact. Okay. So DNA in that plant cell which is eluted out after this homogenization remains intact. Okay. So thereby liquid nitrogen homogenization gives a proper consistency of the homogenate required for DNA extraction. Okay. So first chemical is liquid nitrogen. Now let's see the next chemical, that is most important chemical in the DNA isolation, is the extraction buffer through which the DNA is extracted. Okay, so the next chemical is extraction buffer, which is also called as CTAB extraction buffer. Okay, so this is the most important chemical in this isolation procedure which you have to make. Okay, so this CTAB extraction buffer is having number of chemicals when they are mixed together. Okay, the first and foremost is the cationic detergent, that is CTAB. Okay, so this CTAB detergent is very important in the isolation of CTAB DNA, plant DNA extraction, because the cell wall of plant cell is dissolved by this cationic detergent. Okay, so let's see how much first of all we have to take in this. The first thing that you have to take is CTAB, and the full form of CTAB is cetyl trimethyl ammonium bromide. Okay, ammonium bromide. Okay, so this is the full form of this. This you have to take in 12.5 ml amount. Okay, so first of all you have to take the powder and mix it in distilled water, and the total solution here you have to take is 12.5 ml of CTAB. Okay, so after that the next chemical that you have to take, I have told you the CTAB is a cationic detergent, and its role is to dissolve the cell membrane and break down the cell membrane present in it, and it also removes the polysaccharide. It removes the polysaccharide and dissolves the cell membrane. Okay, so these are the two roles of CTAB here. The next chemical that is used here, see what happens when CTAB dissolves the cell membrane and removes the polysaccharide, all the content of the cellular material or the cellular cytoplasm is exposed. Okay, so there is a presence of DNA also, so we don't want our DNA to get sheared or get destroyed. So in order to maintain the DNA in its native structure, the maintenance of pH of the solution in which the cell is extracted or cell is lysed should be is very important. So for maintenance of pH we are using the next chemical, that is called tris HCl. That is the second chemical, that is tris HCl. We have to take 1 molar tris HCl solution of 10 ml. So in 10 ml solution the concentration should be 1 molar. Okay, so what is the role? It helps in pH maintenance. That is the ideal pH of 7 to 7.2 is maintained here, in which our DNA remains intact. So the role of tris HCl is to maintain the DNA. Okay, so tris HCl we require in 10 ml amount. Okay, in this CTAB extraction buffer. Okay, so the next chemical in CTAB extraction buffer that is to be added is EDTA. Okay, so EDTA full form is ethylene diamine tetra acetic acid. Okay, so ethylene diamine tetraacetic acid, this is a chelating agent which chelates or which traps the ions here. Okay, so what happens here in this, you know, there is an enzyme called as DNase. Okay, that is present in the cytoplasm of the cell. Okay, so what is the role of this DNase enzyme? It degrades the DNA. Okay, whatever the DNA that is present inside the cytoplasm is degraded. Now you have used CTAB to lyse the cell membrane, which even lyses the nuclear membrane also. So DNA is now is in open and is exposed to this enzyme. Okay, so this enzyme will naturally degrade the DNA. Now DNase enzyme, it degrades the DNA with the help of a cofactor, that is magnesium divalent cation, that is Mg2 positive. Okay, so what happens here? This EDTA chelates this Mg2 positive ion or captures this Mg2 positive ion and makes it unavailable for DNase enzyme, and now the DNase enzyme cannot degrade the DNA and our DNA remains intact. Okay, so this is the role of EDTA here, that is it restricts the DNase activity by restricting the Mg2 positive ion. Okay, so this is the third chemical. Now let's see the fourth chemical that is used in CTAB extraction buffer. I'll rub this to write the fourth chemical here. The fourth chemical that is used in CTAB extraction buffer is NaCl, that is sodium chloride. Okay, the next chemical that is used is sodium chloride, and I forgot to tell you, EDTA you have to take in 0.5 ml amount, 0.5 ml, you have to take EDTA as 5 molar concentration of EDTA, that is, and you have to take the solution as 30 ml. Okay, so this much amount you require, that is the EDTA solution that I just told you. Okay, so the next component that is required is NaCl. NaCl is required in 5 molar concentration and 30 ml. Okay, so the role of NaCl here, sodium ion, what it does, you know, the DNA is having the phosphate backbone. Okay, so each phosphate backbone is having negative charge to it. Okay, so due to this, what is happening, the negative charge is repelling the DNA molecule, so DNA molecule is not able to precipitate. Okay, so what happens here, when you add the sodium chloride in solution, this salt dissociates, forming a Na positive ion and Cl negative ion. Okay, so what this Na positive ion does, it binds to the DNA here. Okay, and neutralizes the charge on the DNA, that is positive charge. Okay, so when the DNA charge gets neutralized, DNA tend to get precipitate, or DNA separates out from the solution. Okay, in the mixture where the cell has been lysed, DNA tends to separate out due to the NaCl that we have added in CTAB extraction buffer. Okay, so this is the role of NaCl here. Now, as I told you earlier only, that in plant cell there is number of different plant components such as polyphenols, tannins and all. So such kind of, you know, extra secretion present in plant cell interferes with the DNA isolation. Okay, so for that purpose we add a fifth chemical here, that is called PVP. Its full form is polyvinyl pyrrolidone. Okay, so polyvinyl pyrrolidone, we require this in 10% concentration. So out of this 10%, we require 200 microliter in our solution. So what is this PVP does here? It absorbs all the polyphenols, tannins, etc. Okay, so whatever the extra secreted polyphenols and tannins are absorbed by this PVP, which helps in DNA isolation to us, and protects our DNA also by removing or absorbing all the polyphenols and all that is extracellular components that that interfere with the DNA isolation. Okay, and the last reagent that is used here in the CTAB extraction buffer. So I told you that this is required in 200 microliter amount only. So we require the 10% solution here in the CTAB extraction buffer. Okay, and the last component that is required here is beta-mercaptoethanol. Okay, so beta-mercaptoethanol, it is also called as 2-mercaptoethanol. So it is required in 200 microliter concentration. So it's a strong reducing agent. It's a strong reducing agent. So what it does, it reduces the disulfide bonds. Okay, so it breaks the disulfide bonds present in proteins. Okay, that is present between the cysteine and methionine amino acids. So it breaks the disulfide bond. So due to this, the protein structure gets destabilized. Okay, so destabilization of all the proteins present in the cell helps in DNA isolation. Plus, what it does, it also reduces or breaks the different kinds of tannins and terpenoids that are secondary metabolite. It also reduces the tannins and terpenoids. So therefore this beta-mercaptoethanol is also added in 200 microliter in the said concentration. Okay, so all these different components of this and the last one, sorry, I forgot to mention here, the last one here is distilled water. Obviously all the things has to be mixed in distilled water. So distilled water we have to take here is 40 ml. So when you mix all these components in distilled water, the whole solution becomes CTAB extraction buffer. Okay, so this extraction buffer is later used in the protocol for the CTAB plant DNA extraction. Okay, so this was all about the CTAB extraction buffer. It's different components that are made or that are used for the preparation of CTAB extraction buffer and its role. Okay, so all along with the role I have told you. The next component that is used here is the mixture of chloroform, isoamyl alcohol. Okay, so generally the ratio that you take here is 20:5, 20 for chloroform and 5 for isoamyl alcohol. Chloroform is to isoamyl alcohol, or you can take the ratio as 24:1 also. It depends on how fine or how concentrated DNA fraction you want. Okay, so the role of chloroform here is to remove lipids. Okay, so it absorbs the lipid and forms a separate layer, thereby removing the lipids from our solution. And the role of isoamyl alcohol here is anti-foaming agent. It acts as a anti-foaming agent. Since we have added the CTAB here we have added. So CTAB is a cationic detergent, so naturally it is going to form bubbles in our solution. So to avoid that, this isoamyl alcohol in this mixture prevents the bubble formation, so it acts as an anti-phenol agent. Okay, so now the bubbling can happen due to the CTAB also, and it can happen due to certain phenols that are present in the plant cells. Okay, so this chloroform isoamyl, I have told you that 20:5 or 24:1. Sometimes 1:1 also the ratio is used here, but in some cases it depends on the plant tissue that you are taking for the DNA isolation. Okay, so if you are taking a tissue which is rich in lipid and all, so you have to use the concentration as 1:1. Okay, so it depends on the type of plant tissue that you are using for the DNA isolation. Okay, so thereby you adjust the concentration likewise. Okay, and the fourth reagent that is used here is either isopropanol or ethanol. Okay, so generally the first precipitation done is here is with 100% isopropanol or ethanol, and the subsequent washes is done by 70% ethanol. The thing is, you have to take this component as chilled. Okay, it should be ice cold, because the precipitation is best achieved when this reagent, that is isopropanol or ethanol, is in ice cold condition. Okay, so what this isopropanol and ethanol does, it helps in DNA precipitation. Okay, it helps in DNA precipitation, and then it is a, how it does, so it is a dehydrating agent. See, both of them is dehydrating. Okay, both of them are dehydrating agent. Either of the one you can use. They are dehydrating agent. Means what they do, they remove the water molecule from any biological biomolecule. Okay, so in DNA also it removes the water molecule from the surface of the DNA. There is no water molecule left due to the alcohol here. So the DNA gets hydrophobic, and due to it it gets precipitated in the solution. Okay, so this is the precipitation solution. The ideal condition for precipitation is -20 degrees centigrade. So here the incubation is done at -20 degrees centigrade. Okay, so this is the role of isopropanol and ethanol. And the next component or next chemical that is used here in the CTAB DNA extraction along with its role is the TE buffer. TE buffer. TE buffer means tris HCl plus EDTA. Okay, so it is tris EDTA. Okay, TE means tris EDTA. So tris HCl, it is used for maintenance of pH, and EDTA is chelation of Mg2 positive ions. Okay, so what this buffer, why it is used? See, first of all, pH should be maintained, and EDTA, there is no Mg2 positive ions that should be available in that solution, so that our DNA remains intact. Okay, so this buffer is used as a solubilizing buffer for DNA, or DNA is stored in this buffer. Okay, and you can use the DNA by dissolving in this buffer. Okay, so this is required at the last, where the DNA is formed or DNA is isolated in a purified form, and you have to now dissolve the DNA, so it is used that is this TA buffer. Okay, so the last chemical that is used here are two enzymes, that is RNase and protease or proteinase K. Okay, so these are the two enzymes that can be used in either 3 microliter or 5 microliter concentration. Okay, you can use 3 microliter also, both of these enzymes. If the protein fraction is present in that particular plant cell is more, then you can use proteinase K and RNase enzymes for degrading the RNA. So RNase degrades the RNA, proteinase K degrades the protein. Okay, so what happens here in this, both of them are required when you want a high quality of DNA or when you don't want any kind of contamination in your DNA. Okay, so then at the last, after the ethanol wash, you use RNase and proteinase K enzymes, that too in 3 microliter concentration, so that whatever the contamination of RNA and protein is there is removed by this. Okay, so these are all the chemicals that is used in CTAB extraction buffer. Now we are going to see the procedure. All the different chemicals, that is the amount and all, means the amount in which they are required and all, will be given to you in the description. You can refer the description for the stepwise protocol for the CTAB DNA extraction. Here I will tell you the protocol in a flowchart manner so that you can get it. Okay, so for the exact amount of chemical that is to be added in a particular isolation tube, you can refer the description below. So now let's see the procedure for the CTAB DNA extraction. Okay, so first step obviously in CTAB DNA extraction is homogenization. Okay, so homogenization by liquid nitrogen. Okay, so when you use liquid nitrogen for the homogenization of the DNA, so when you get a fine fine textured DNA, then you add the extraction buffer. Okay, extraction buffer. Okay, the CTAB extraction buffer you add. Okay, so after adding the CTAB extraction buffer, what you have to do, you have to mix it well. All the grinded material, all the homogenate is properly mixed in CTAB extraction buffer and it is incubated at 37 degree, or you can incubate it at room temperature also. Ideally it is kept overnight for to bring about the proper extraction of the DNA. You can go for either 3 hours also if you want. So it is incubated in the CTAB extraction buffer. So afterwards you can keep it for overnight. Okay, so if you are keeping it for overnight, then day after you have to add chloroform, isoamyl alcohol. Okay, I told you 3 ratios, either it can be 20:5 or it can be 24:1 or it can be 1:1. See, it depends on the what type of DNA sample you are isolating. Okay, so you have to add the chloroform isoamyl alcohol ratio, and you have to add in equal concentrations. For example, if this component here is 0.5 ml, then you have to add this 0.5 ml. Okay, so it should be equal in amount. Okay, so after that you have to mix it well by inversion. Okay, inversion means what? To and fro motion like this. You have to mix the tube. Okay, so once you mix the tube here, what will happen? It will form a homogeneous solution. So after that there is a centrifugation. So you have to centrifuge. Next step is centrifugation. So you have to centrifuge this at 10,000 RPM for minimum 10 minutes. Okay, so when you do this centrifugation, let me show you here. There will be 3 different layers. This is the central layer where different kinds of all protein debris or the cellular part will be there. The below layer here will be of chloroform, which will have all kinds of lipids, and proteins will settle at the center. And the top aqueous layer here, the top aqueous layer will be having the DNA. Okay, so this is the centrifuge tube that I have shown you just to explain you. So you have to take this aqueous layer in the new tube. Okay, so in the new tube, carefully using the micropipette, you have to remove this aqueous layer or take out this aqueous layer into the new tube, which is RNase free. Okay, so you have to take this aqueous layer like this. Okay, so when you take this in the new tube, now you have to do the further treatment with the new tube and discard this one. Okay, so this is the phase separation step that has occurred here. First of all, the first step is cell lysis till here. Now the phase separation occurs. So the aqueous phase is taken in new tube and we have to go for next step, that is precipitation. Okay, so you have to add isopropanol or ethanol in equal amount. Okay, so for example, the aqueous phase you recovered from the previous tube is about, suppose, 5.5 ml. So you have to add this in equal amount. Okay, and should be chilled. It should be done in chilling condition, and the reagent, that is ethanol or chloroform, should also be in a chilling condition. Okay, so now what happens when you put this chilling condition? Immediately precipitation starts. Okay, so now when the precipitation starts, you can keep it for either 3 hours, or if you want proper precipitation, you can go overnight. So it depends on the type of or amount of DNA that is present in the sample. You can keep it overnight also to get a proper yield. Okay, so on the next day, what you have to do? You have to centrifuge. Okay, so you get pellet that is of DNA. Okay, you get pellet that is of DNA. Okay, so now the protocol ends here, but for proper more purified or more purified form of DNA, what you have to do, when you get the DNA in the isopropanol precipitation, then subsequently you have to give the washes of 70% ethanol. 70% ethanol, what it will do, it will remove all the different reagents that are present or traces of the reagent present from the DNA sample, and then when you can go for either 2 to 3 washes of the 70% ethanol. So at last, after the last wash, keep the tubes in open and allow the remaining ethanol to evaporate, leaving the DNA inside the tube. Okay, so now what you can do, this DNA can be dissolved in TE buffer and then it can be used. So generally 1x TE buffer you use for the storing of DNA, and then this DNA that is dissolved in TE buffer can be used for different kinds of analysis such as RAPD analysis, and restriction digestion or PCR, etc. So you can do different kinds of analysis using the extracted plant DNA. Okay, so this was all about the the whole protocol for the DNA isolation, that is CTAB plant DNA isolation protocol. Generally, see, the ideal sample is very young leaves. Okay, you have to take very young leaves, or you can take young stem also, but remember, don't take venations in that, or don't take a hard tissue. Young leaves are soft, and you can get the DNA in large amount because they are in multiplying stage. Okay, so this is the whole protocol. The exact the amount of solutions and all you can get in description. The whole protocol is there in description. So I think I hope you have understood the whole protocol that I have explained to you with the role of each and every chemical. So thank you for watching this video. If you like the video, please press like button, share, comment and subscribe the channel. Thank you for watching.